My name is Isa,
I'm Hannah.
And I'm Cindy.
and we want to thank you for taking the time to watch our presentation. COVID-19 is a disease caused by SARS-CoV-2 that has devastated the world for over a year, and much is unknown about how this pandemic might have started. We did a comparative analysis for the three protein spike, NSP5 and NSP12, found in the human coronavirus SARS-2 proteome. The study looked at what bearing the spike protein NSP5 and NSP12 might have had in creating the current pandemic by looking at protein characteristics known to alter protein capability, leading to aberrance.
Our data set was based on the best BLAST-P hits generated from the NCBI database. Figtree software compared the aligned sequences for visualization of an SH-like phylogenetic tree. We then used three prediction algorithms, ScanSite, NetFOS, and NGLide to find motifs, phosphorylation, and glycosylation sites respectively. AliView was used to calculate conservation scores for all the post-translational modification, and PyMo helped create a graphical representation of these features. Lastly, Spider software was used to perform statistical analysis using Python.
The phosphorylation sites in the spike protein displayed a significant difference between the SARS and MERS clade. Multiple sites for serine and threonine residues exhibited very low conservation scores. Phosphorylation site 50 had a conservation score of 3.5%, making it a distinct site in SARS-CoV-2. The data for motifs showed significance between the SARS and outgroup clades. Spike is highly conserved in N-glycosylation sites with no significant difference between clades. Mutations or post-translational modifications to serine-rich phosphorylation sites may play a role in the conformational changes expressed in the spike protein, while a decrease in furin cleavage efficiency resulting from a mutation in motifs may have possibly led to the prevalence of zoonotic transmission and pathogenicity of the virus.
Post-translational modifications were 100% conserved in the SARS clades for NSP5, also known as a 3CL protease. Such a level of conservation highlights the importance of maintaining the specific function to cleave the replicase polyprotein. Scientists can utilize this information to develop protease inhibitors that are effective for other target sites within the SARS clade dataset.
Phosphorylation sites shared between the SARS and outgroup, and in both outgroups were found to be statistically significant in NSP12. Likewise, the motifs shared between the SARS and outgroup and MERS and outgroup clades were found to be significant. For all 24 sequences, the conservation of phosphorylation sites ranged from 17 to 100%. The SARS clade was found to be 100% conserved. SARS-CoV-2 generated 7 motifs from its scan site results, with conservation scores ranging from 13 to 100%, which was predominantly conserved.
These findings suggest potential reasons as to why COVID-19 has had such a large impact on society, and also highlights ways that we can create effective therapies to fight the virus.